All right, so first up, someone asked, had sent me an email yesterday, I think, about some of the homework. And in particular, it was the homework from chapter 5, I believe. And it was a question about lipids. And I'm just going to kind of get to the general lipid portion here and see if I can find the question they were talking about. I believe they asked about a phospholipid. Someone said there was a question about a phospholipid, and it asked how many fatty acids in the phospholipid, and they answered 2, and it was marked incorrect. So I'm just curious if it was actually a phospholipid or if it just said a triglyceride. So I'm trying to, and if any of you had that same issue, you see here at the bottom of each question, oh, crap. Sorry. What happened to my meeting thing? Really? Sorry for that, I clicked the wrong button. What I wanted to tell you was anytime you have any issues with the questions, I really encourage you to hit this button at the, let me get all the way back there, and then I'll share the screen with you again. I guess you can see it now. Excuse me.
I had that issue as well. I just used my better judgment, but moving forward, I'll press the feedback.
Yeah, send them a feedback because they update. I have a list of when they update things. So the next update is February 20th. So anytime you have issues like that, click that button and just send them an email and someone will answer you. I believe someone answers you. It must have been on this one with the figure walkthrough of the phospholipid. And how many fatty acids are in a phospholipid? All right, so let's just answer that too, because that's correct. And now it says correct. It must have known that it was me. Do you guys see that? It said how many are in a phospholipid, and I clicked 2, and it said 2 fatty acids are correct. So they've probably gotten a lot of feedback already and fixed the answer. Check your score and see if it went up for that question. Let me look. Chapter 5, phospholipid walkthrough. Was that the place? Do you guys remember?
No, I was wrong. That was not my issue. I'm going to read through the questions really fast and tell you where I was caught off guard. I should have jotted it down.
Is it in this? Is it in this one or no?
Not in that specific section, but it is in chapter 5.
All right, let me go back then. Yeah. Well, I'm the instructor, so it doesn't matter if I get it wrong. All right, let me know when you find it. And sort of in general to the rest of you, again, I would encourage you to push those buttons and ask them and for feedback. And it looks like, let's see, when I go back to mastering, it looks like most of you have already completed that one. So at least that's what I thought I saw. I should be able to amend those, although it's like it's not even a part of a percentage point if it's just one question. But if you can tell me specifically where it is. And for me, that marked correct. So I don't know. All right, so other than that, let me hit this stop share. Other than that, questions that you have for the pending doom tomorrow of the first lecture quiz 1 through 5, right? So other questions that you have, the lab kits, a lot of people have already picked them up. Please go pick them up. If you truly, I've been told that there's no way for the college to mail them to you, the bookstore or the college. I'm still trying to pursue that. if you really, really, really live out of state and you cannot pick up the lab kit, you do need to send me an email so I can give the administration specific names and student ID numbers of students who are affected by this policy that they've enacted. So I know that in the fall, the students paid for their lab kits. The wards kit, which you guys are purchasing, is which you guys are getting for free, is $150 kit for each student. And I know that because students were actually paying for them, the bookstore was able to arrange shipping. So I don't know what the status is, but believe me, I am sending the emails all the way up the chain to all of the people who actually, the administrators, not the president, because he doesn't have actually the power to do this, but so the administrators who are in this chain who would be able to help us with this policy of the bookstore not being able to ship it. So please, please, please just go pick up your kit. I gave you the bookstore hours. I know that the evening hours are going to end this week. I don't know what day this week. I would call the bookstore, and if that arrangement has been made with security, the bookstore will have been informed. And when I get that email as well from the chair of my department, I will pass that on to you guys. But right now, as many of you as possible, please just go pick up your kits at the bookstore when they're open. Again, I would take your ID, your student ID, and a driver's license, just because, you know, I'm a parent myself, and, you know, something I always plan, hope for the best, plan for the worst, right? So I would take identification with you. I would have your registration up on your phone just in case that they're like, oh, you're not registered for this class, because that's what they're going to do. They're going to check your registration, and then they're going to just give you the lab kit. So most likely they'll just ask for your name, but I don't know. I'm not in the bookstore. I don't work there. I'm getting all of this second and third hand. So please pick up your lab kits. The first lab I have is due this Friday. Don't make a due date, you won't do it. We procrastinate by nature. That's a soft deadline, meaning there will be no penalty, and I've left that lab available until Monday. There is not a place yet for you to submit your results from the experiment because I'm going to add a little bit to the lab report. Those of you that already have the kits, you will realize that there is a data table and questions, and then on the back there are the answers to all the questions. So I'm going to be changing that so that there's something that you have to actually think about before you type out and just copy the answers from the answer key. So that'll be popping up sometime during the week this week. If you are excited about doing the lab and you want to get it done, by all means, watch the lab video, complete the lab exercise, and then take a photograph of yourself with your results, with at least some of your results. I think you use a spot plate for this one. So a lot of students just sent me a photograph of their spot plate with themselves, right? I need a picture of you actually doing the lab to verify that you've actually gotten the kit and are actually doing the work. That will be your first upload for this lab. And then the second one, there'll be 2 questions in the lab. The first one will be to upload your picture, and the second one will be for you to upload a document that will be the actual lab report. It most likely will be a fill in the blank type of lab report. So there'll be, just like in the lab manual, the lab booklet, there'll be things to fill in as you go along, colors, I believe, and then there'll be questions for you to answer. So that's what's going on with lab. Please try to get your kits as soon as possible. I have a second lab due on the 19th as well. We're going to do 1 a week for the last 3 weeks of February. So it is important that you get those kits as soon as possible. Again, if you are someone who really lives out of state and you cannot actually physically come pick up the lab kit, you need to get me your name and student ID number as soon as possible so that I can use that as specific examples of students who can't complete the course because they can't get their lab kit. And I'm sure they'll come up with some system of shipping them to you, but they're not going to ship them they're not going to ship them anywhere in New Jersey. So you're going to, if you live in New Jersey, you're going to have to come pick it up. Okay, so back to questions about the pending doom tomorrow. Yes, no, maybe so. Nothing, really?
I do. Okay. I have, sorry, I just had to wait for silence at my house. I'm just getting slightly confused with the different types of purines. It's just when I look at them, I'm like, what is supposed to, which one goes in specific, that homework where it wanted us to, it took me a long time to figure out what needed to be put into the correct category.
Yeah, that happens sometimes in those homeworks when they have like all the little itty bitty details.
Yeah.
So a purine, what I always tell students to remember is a purine, short word, long picture. So a purine has 2 nitrogen containing rings. Plus, right, if it's a nitrogenous base, then it's going to have, that's a purine. A purine is 2 nitrogen containing rings. The ones that we're focused on are adenine and guanine, right? A and G, those are the ones that are involved in DNA and RNA. There are other purines in the world, right? We just aren't talking about all of them. If it's actually a nitrogenous base, right? If it's a nucleotide, right? Or a nucleoside. So the nucleoside is when it has the sugar attached. You can just remember S for sugar. The nucleotide is when it has the sugar and the phosphate attached, right? So we refer to those as, so a nucleotide is this nitrogenous base plus, I should put an equals there, equals, nitrogenous base is the nucleoside plus phosphate. I don't think I can super subscript when I'm in this weird little whiteboard area, but so the nitrite, so, and then when you put those nitrogenous bases in a chain, that's when it becomes a nucleic acid. That's technically the chain. Your book, I think, calls them polynucleotides. No one uses that word ever. That will not be on the test because no one ever uses that word, right? And then, of course, that's same. So nucleic acid is a chain of polynucleotides or a chain of nucleotides. And then, of course, the choices for you for nucleic acid right now are just going to be DNA or RNA. And we're not going to talk about all the specific different types of RNA right now. We'll wait and hold on that until we get to chapter 17, and then we'll talk about messenger RNA and small nuclear RNAs and all these different types of RNAs that are out there in the world. Better or worse?
Much better. And then I have another question as well.
Great.
Can you go over anti-parallel?
Oh, absolutely.
Thank you.
So I may have to actually draw for that. So anti-parallel, the DNA is anti-parallel. What that means is to do with this. So I'm going to draw a chain of DNA like this and like this, right? To make it look like a double helix. Now, what anti-parallel means is that one end of the chain, we call it the 5' end. So these are the nucleotides across here, right? So the bases are in the middle, the phosphates and the sugars form the string, the black string that I drew, right? One end of a chain of DNA, we call it the 5' end, and the other end of that same chain we refer to as the 3' end. And that has to do with what the chemistry is at the end of the chain. And this is going to be important for later when we talk in chapter 16 about replication. But if we follow this, chain right here, right, all the way to the bottom, 5' end, that's going to mean that this other end of the chain is a 3' end, we call it. And again, that has to do with what's on the ends of the chain and why DNA is called anti-parallel and what gives it its, what actually makes it a double helix or a helical structure that turns with this wide turn and then a skinny turn is the anti-parallel nature of the strands, because the complementary strand has the opposite orientation. So the complementary strand has a 3' end across from the 5' end, and then down here would be a 5' end. And for right now, that's all you need to know about that. You don't need to know the chemistry. But we will talk about the chemistry of what makes 5' different from 3' again when we get to chapter 16. Super briefly, I can just tell you that the 5' end, or sorry, the 3' end has a hydroxyl group on the 3rd carbon of the last deoxyribose. The 5' end has a phosphate on the 5th carbon and the 3rd carbon. So I'm just going to put a PO4 out here. I'll just put P out there. And if I had drawn this picture better, like it is in a textbook, right? They always end this strand so that it doesn't look like the 5s are on the same side. But alas, you're stuck with my bad drawing. All right, what's important is this part here. 5', the complementary strand is 3'. They're anti-parallel, meaning it's not 5', 5'. Right? It wouldn't be able to form a helix if both the chains were like this. Those 5'. Nope, that does not happen. Right. Did they ask you that in the homework, or was it just something that was on the sheet of the characteristics of DNA?
I believe, hang on one second. I believe the question, the question was, it gave us like 5' end, AT, 3' M, whichever the sequence is, and we had to give the complementary.
Oh, okay, right. And then you just have to be sure to put the complementary end on it, right? So for example,
perfect.
always DNA, we describe it, the 5' end is always on the left or on the top if the sequence is horizontal. So for example, right, if it was 8, caps lock on here, A, T, C, G, G, A, whatever, ... right? Then the complementary strand has to be 3'. And then we're going to have complementary bases. They're assuming that you've had, you know, enough biology before to know that the A pairs with T and C pairs with T. That's like a precursor. I don't think there's anything that specific on the quiz. I mean, I know there isn't anything that specific. I'm not going to ask you which purine is complementary to which pyrimidine. I just want you to know that a purine always pairs with a pyrimidine. Okay, and I would not even expect you at this point to be able to tell me that the purines are adenine and guanine. I would expect you to be able to look at a picture of a purine and see that it has 2 nitrogen-containing rings and say, ah, that one's a purine, right? That's part of a nucleic acid. And the one with a single nitrogenous ring, you should be able to say, oh, that's a pyrimidine. But again, I would not expect you to know this yet.
Thank you.
Yep. Anything else? Let me put something in the chat, but I can't make my cursor go there. Let's see. Can I just, no, well, let me minimize that either. Gotcha. Amino acid structure, please. Okay, so amino acids, there are 20 of them, right? So so proteins, long chains of amino acids, which I will forever abbreviate like that, right? Amino acid, and you need, we need to remember that for the rest of the semester. Yeah, if I can do it correctly, there we go. There are 20 different amino acids. That's that picture in your textbook on whatever page that is. Just scan real fast. So that's one of the big tables in chapter 5. I would not expect you to be able to tell me the names of the 20 amino acids. I would not expect you to be able to draw any of the 20 amino acids. Again, the expectation is if you see a picture, a chemical picture, like in your book, you would be able to tell me, oh, that's an amino acid, not a nucleic acid, not a nitrogenous base, not a purine, not a carbohydrate, not a lipid, not glycerol, right? Not a steroid, not a phospholipid. So what's different between all the 20 amino acids are what we call their side chains or their R group, which also is known as, for many of them, is part of what we would call their functional group. That probably is not chemically correct, but that's how I was taught, that's where often the chemistry happens between amino acids. That's the reason they're called the functional group. And those side chains, there are 20 different side chains. That's what makes the 20 different amino acids. Excuse me. Now, every single amino acid, what they all have in common is their basic structure going across. And for that, I probably will try to draw that rather than type it. So for that, we are looking at always, 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 there is a nitrogen with 2 hydrogens bound to it. And that's because you'll either see it with 2 or 3, right? So the basic formula for our nucleic acid, for our amino acid, is going to be like this, H3. If we're writing it just in a sentence, like just straight across, H3N, right? A nitrogen with 3 hydrogens bound to it, carbon, and then a second carbon. And that second carbon typically belongs to a carboxyl group, which is why I wanted you to memorize those groups from chapter 4. A carboxyl group, remember, has a carbon with sharing 2 electrons with an oxygen. And then remember, carbon has 4. It would like to share 4 electrons. So 1, 2 is sharing with this other carbon. And then lastly, with a hydroxyl group here. Right? So when I say, if you've watched the videos and I say, look for nitrogen, carbon, carbon, look for an amine group, carbon, and then a carboxyl group, that's what I'm trying to get you to see when you look at a picture and say, oh, is this amino acid, right? So this is the amine group here at the beginning. This is that carbon, so an amine group, nitrogen, carbon, carbon. This is the carboxyl group here. Right? That's why they're called amino acids, because of the amine group, right? Again, that was one of the groups, the functional groups from chapter 4 that I asked you to memorize. So if I'm drawing out the whole amino acid here, I've got nitrogen with its 3 hydrogens attached, and then a chemical bond. Nitrogen would like to share 5 electrons. It only has 3 in its outer shell. And then here's a carbon. And this carbon is going to share or have 4 chemical bonds attached to it eventually, right? I'm just doing the backbone right now. And then we have the second carbon that's actually a carboxyl group. And that's always the same. Nitrogen, carbon, carboxyl, nitrogen, carbon, carbon, nitrogen, carbon, carboxyl, right? When you put these amino acids in a chain, what's going to happen is it's going to be a dehydration synthesis reaction, and we're going to lose water. And where the water is going to come from is here. One of these oxygens, this hydrogen, and one of the hydrogens from the next amino acid in the chain. All right, so that's going to be one of those dehydration synthesis reactions that was mentioned at the very beginning of the video for the first video for chapter 5. Put them in a chain. Now, but back to the general structure. Everything that you see that's in color in your textbook, they've got yellow ones, green ones, pink ones, and blue ones. So let's make it pink, right? And they've put the functional groups all on top. So I'm just going to draw a square here and put an R in there for functional group. And that's what all the 20 different side chains are. That's the 20 different R groups. And then at the bottom, there's always just a hydrogen hanging there. And people draw these either way. In real life, this is a three-dimensional thing, right? So the R group and this hydrogen literally can spin around the carbon. So a lot of textbooks draw them in reverse. They put the hydrogen on the top and hang all the functional groups from the bottom. It does not matter. Same thing. So what makes alanine alanine, for example, is because the R group for alanine is CH3. So here, instead of just an R, we would put, I'm gonna erase this, see if I can put something in there actually, maybe. Inside of there, for alanine, for example, I would have to draw another carbon. And then your textbook shorthands it by just writing CH3. But if I were drawing all the bonds, right, I would draw all three of the hydrogens attached to that carbon. Like that, right? So carbon's sharing 4 electrons, and this is the functional group, right? This is what makes this particular amino acid alanine. What makes a different one tyrosine? It's because it has a different functional group. It's these functional groups that are interacting in the secondary, tertiary, quaternary structure of polypeptides that actually turns them into functional proteins, okay? Pretty much the only thing that is functional as a peptide, they're functional basically in 2 ways. Peptides are functional as signaling molecules to like get a protein to a particular location, and then a lot of times they're chopped off. And peptides themselves are, most of your hormones are peptides. So they're short sequences of amino acids, maybe 20, 25 amino acids or less or fewer that actually tell cells. Hormones are things that send signals to cells to tell them what to do. And most of your hormones are actually what we would call peptide hormones, which you will learn lots about when you go to anatomy and physiology. All right, Bo, better or worse? Do you need more or is that good? You are welcome. And everybody needed that, I would guess, right? Anything else? I'm kind of looking at the end of the chapters, see what they're asking you about. Not much, and then it's time to go on to the cells. All right, other questions, comments, worries, concerns? Just as a reminder, right, the test is multiple choice, and there will be pictures, there will be drawings, better than this, there will be drawings of organic molecules that you will need to identify the structure and identify amongst the choices what the function of that structure is. Oh, carboxyl carbonyl, that's not super important.
I second that. As soon as she typed it, I was like, oh, yes, yes.
Right, that's why it's important you guys ask those questions that you're thinking about in the back of your mind, because everyone has the same, right? So the carbonyl is a group. So, and this is the carboxyl here. Carboxyl, oxyl, because there's a hydroxyl on it. And then a carbonyl group, if I remember correctly, is just the carbon double bonded to the oxygen. And this bond is left blank, right? Which means it's usually a hydrogen, right? So carbonyl, no hydroxyl group. Carboxyl, for me, when I look at a picture and I'm trying to figure out which of the 4 groups it belongs to, literally I look for a backbone, if it's going to be a protein, of nitrogen, carbon, carbon, double bond oxygen, nitrogen, carbon, carbon, double bond oxygen. Right, so like if we look, pen there to hold place, but I'm trying to see this picture here. Figure 515. Let's see if I can get us to that without much ado. Sorry. All right, let's see if I can get us to just looking at the picture in the book. Sometimes this doesn't work so well early in the morning because a lot, they're like, oh, who are you? Okay, open my course. Because everyone in the whole country is trying to open their Biology 1 courses and use mastering at this moment, right? So trying to think which is the easiest. Sometimes if I click on this, it says you don't have an e-text. Oh, today I do, okay. And it's tough. I don't think Pearson lets us have a student view. Connect does. So sometimes I can't see what you see. So let's see, menu, contents, water, 4, 5. Okay, so inside chapter 5, I want here, section 5.4. And again, this will probably take a little bit to load. So I'm gonna try to be slow, go slowly. I don't want all those. I just want that picture. That one. Oh, it's coming. All right. There's all the 20 amino acids. And then I want the picture after that. 5.15 polypeptides. This picture here. Right. So this is the picture that I was trying to roughly draw with those that I'm saying there was a dehydration synthesis reaction to form a peptide bond. This bond is important. This bond, it's called a peptide bond. It's going to come back to haunt you again, but later, right? All of these molecules, we're going to keep talking about them all semester. This one, so this is showing you what we would call a tripeptide, right? Or just a peptide. And you can see that every time we put another amino acid on the chain, we're breaking out a water molecule. The oxyl of the hydroxyl is being joined to a hydrogen from the next amino acid from the amine group to form water. So when I'm looking at a molecule that's in a long chain and I'm trying to decide, is this a lipid? Is this a carbohydrate? Is this a protein? Is it DNA or RNA? This is what I look for when I see the pattern of nitrogen, carbon, carbon, double bond O, nitrogen, carbon, carbon, double bond O in the backbone. When I see that repeating over and over again, I'm like, oh, protein, right? These are the R groups. They're all different. I'm not looking at those. I don't care what those are. I'm looking at what is the same, what is the repeating structure, and it's this. Nitrogen, carbon, carbon, double bond O. Nitrogen, carbon, carbon, double bond oxygen. Nitrogen, carbon, carbon, double bond oxygen, right? To break these peptide bonds, if I'm digesting protein, for example, which we'll talk about in chapter 9 or 10, 9, I think, right? Then I'm going to have to add water back to break this bond. And that's going to reconstitute the hydrogen on this amine group and give the hydroxyl group back to this ending amino acid. So notice that this last amino acid here, this single amino acid, right? It still has its hydroxyl group because there's not another one being joined on yet. That's important. Of course. All right, so other questions about that before I make the textbook go away? No, I'll just leave it there. I'll just stop the sharing there. Yes, no, maybe so. You sure? I'm waiting because I don't want to stop the recording and then have you guys ask me more questions. And then the students who haven't been able to come to the Zoom meeting don't get to hear the answers to those questions. So I'm just kind of giving you a minute to think about it. You'll have all day to take the test, right? It starts at, it'll open up tonight at 12:01 a.m., right? So the first minute of tomorrow, and it'll be open until 11:59. Please don't wait until 11 o'clock or 10:30 to sit down and take the quiz because a lot of students are on Canvas at night and do their homework at night or whatever they need to do. So you don't want to be in the highest traffic time. And other than that, like I said, 45 seconds a question, so it goes really quickly. So you don't have time to look up the answers. So you need to be studying enough today, especially so that you'll be able to just read the question and be like, okay, I know what that is, and just go ahead and answer it. If you try to look up every question, I understand taking tests online, there's this, you think that, oh, I can just look them up. I'll just look up the answers. It'll be okay. You don't have enough time. Okay. And that's not the point. The point is it's a test. It's not a homework assignment. That's what you've been doing and mastering where you can look up the answers. This is a test, right? And like I said, when we go back in person in the spring, in the fall, hopefully, you're going to have to sit down and take tests again in person. So it's important, and they're timed, right? Because we only have an hour of lecture, or we have an hour and 15 minutes of lecture, and that's the amount of time you have to do the test. So it can be done. You know, people have been taking this particular test or a similar test, not this one exactly, but similar online tests that I've been constructing since the spring. And people get A's, people get B's, people get C's, people get D's, people get F's, right? So it's the same. So try to study so that you're ready for the questions. That's the best I can tell you. I would review your homeworks. You can go over those after you've turned them in. They're always available for you to look at again. And obviously study the notes from the note outlines. And hopefully you've watched the videos, the lecture videos, so that you know sort of the vocabulary I use and the words that I put together, because it's the same way that the test is going to be, the lecture quiz is going to be. It's going to be I don't use questions from the test bank. So if you printed out all of the answers to all of the questions from mastering, that's not really going to help you, except if you want to use it as a study tool, because I'm not using those questions, right? Someone, and not in Bio 1, but someone in microbiology told me they were copying out all the Connect questions and the correct answers. And I was just like, I don't use those questions on the test. I told you that. So no, she's like, is that a good way to study? And I'm like, no, study the notes. And obviously for this particular one, you do have to be able to recognize the pictures of organic molecules and keep a list of their functions in your head, right? All right, other questions, worries, concerns? No? All right, if not, then I will see you on Wednesday and we'll be ready to talk about chapter 6. Try to get your lab kits either today, tomorrow, or Wednesday, whenever. The lab itself, I think, doesn't take very long, maybe an hour. And you should watch Professor Ramirez Wright's video before you try to do the lab. And again, worst case scenario, if I don't get the lab report form loaded by tomorrow or before Friday, all you have to, the most important thing is to take a picture, a photo of yourself that can be saved and then uploaded with your lab report of you with the data, with the results. I mean, right? And then the form for you to fill out for the lab report, you can always do that later. All righty, then I will see you on Wednesday. If you think of something that you really have to ask me, remember I have office hours today at 11. So find that find that announcement, and there's a the passcode always for my office hours is just my last name. It's just Stuart, and the meeting ID for Monday office hours is, I can give it to you right now in case you can't find it. Monday office hours, I'll just type it on one of these pages. Hey. All right, so office hour meeting ID is for Mondays is 945-0457-8150. And the passcode is always just Stuart. Right? At 11, if you, like I said, if you think of more questions or something that you forgot to ask me and you're just like, oh, I'm not gonna be able to sleep tonight if I don't know, just come to the office hour, 11 to 12. Ask me then. All right. Other worries, concerns? Yes, no? All right, then I will see you on Wednesday. Bye.
Thank you.
You're welcome. Good luck. Study, study.